in this module, we will talk about microbial growth kinetics. That is the continuation of the second chapter of fermentation. As we know that fermentation is the mass culturing of the organism, especially microorganism, for the sake of any desired product. So, when there is a mass culture, then there is a growth. So, in this chapter and in the different modules, we will talk about that what kind of the changes occur during the mass culturing. So, first we should know about what is the growth. Growth is a very simple term that refers to any change in the state, in terms of number, size, volume, or in any sense. So, in terms of microbial growth, basically the growth is the ratio between the birth and the death. So, as concerned the growth kinetics, so as we know that the growth is the ratio between the birth and the death, and the kinetics is the growth with respect to the time, how it changes with respect to the time. So, in short, we can say that change in number of the cells, change in the size of the cells with respect to time. So, when we talk about the change with respect to time, then in short, we are talking about the microbial or a growth kinetics. As concerned the growth kinetics, keep in mind the growth is further divided into 4 different phases in general talk. Lag phase, log or exponential phase, stationary phase, death or we can say decline phase. As I already told you that the growth is the ratio between birth B and the death D. So, as concerned the lag phase, when the ratio between birth and the death will be 0, then we called as lag phase. But when it more than 1, then the growth will be in a log or exponential phase. But when this ratio become 1, and then it can be called as stationary phase. But when this ratio decrease than 1, it means death increase than the birth, then that we refer to as decline phase. So, in next slides, we will talk about all these phases in detail. As concerned the lag phase is the period of adaptation for the cells to their new environment. So, in the start, when we inoculate the fermented vessel, in the start time, the cells try to acclimatize in the new environment, and then there is no increase in the cell number. So, no change in the number, but they can change in their volume a little bit. We can say that there is a change in the cell mass. So, as concerned the log phase or exponential phase, in this phase the cells have adjusted to their new environment. Because they have already adjusted their environment in the lag phase, then they are ready to multiply. We know that when we talk about the cell cycle, then there is the need of some cell constituents before division. So, when there is a M stage, so when we talk about the log phase or exponential phase, then all the cells are ready to go in M stage, which the cell can be divided either mitotically or meiotically. So, in this phase, the cell have adjusted their new environment and they are ready to multiply exponentially. So, this can also be called as balanced growth, in which all the components of the cell grow at the same time. So, the growth rate is independent of the nutrients. So, in this phase, almost all the nutrients are in excess amount. So, the third stage, which we called as stationary phase, with the exhaustion of the nutrients, because when there is an exponential phase, then there is the maximum utilization of the substrate. But during this process, when there is any deficiency in the nutrients, then there is effect on the birth or in the mitotical divisions of the cells. So then there is a stage when we can say that the ratio between the birth and the death become equal. So at that stage we can call as the stationary phase. So in this stage cells remain active, their metabolism is mostly active, but this is the stage when their secondary metabolites are produced. In this diagram you can say the different phases: lag phase, which we call as the first stage, and then is the exponential phase, and then a stationary phase, and the last, when we say this, a decline phase. In the next slide we will see that what is the decline phase. So the decline phase, in which the living organism population decrease with time due to the lack of the nutrient, either by some toxic secretion secreted by the cell itself. So as I have already told that in the decline phase the death rate exceeds from the birth rate.